Hello, thank you everyone for joining the TMACOG Water and Wastewater Operator Training for 2022. We're going to allow for just a couple more minutes for everyone to get connected and to address any technical issues. I'd also like to remind everyone that in order to receive operator credits, each attendee must be separately registered for the webinar. Also, please take a moment to download the contact hour request form from the handouts section of the GoToWebinar tooltip. And please also note to close out any other screens or applications besides GoToWebinar to assure that your participation is correctly recorded. Thank you, and we'll begin shortly. Hello, everyone. We're going to go ahead and get started with the webinar training session for today. My name is Matthew Kennedy, and I'm a water quality planner here at TMACOG. I'm also joined by another TMACOG water quality staff member, Eric Kostecki. Eric will be administering the polls and taking questions from the speakers. I'd like to give a big thank you to Jones and Henry Engineers for their sponsorship once again of this year's webinar series, and to our presenters for their time in preparing this valuable training. Today's agenda will include presentations on wastewater monitoring to track and respond to disease outbreaks by James Boyd with Greene County Sanitary Engineers, Dr. Susanna Bararova, and Judith Strathoff of The Ohio State University, as well as a presentation entitled, Hey, Small Plants Need Dewatering Too by Daniel Miller from Jones and Henry. Before we get started, I'd like to provide a short overview of the webinar format today. All of the attendees will be muted, but we encourage you to participate in discussion using the question function on the GoToWebinar toolbox. You can find the questions not answered during this webinar will be addressed following. You will also be asked to participate in a total of 8 polls during this webinar, 4 for each presentation. It's important that you participate in these polls because they help our presenters get to know you and they count towards your webinar participation score. More on this participation score in a moment. You'll be asked to participate in a survey at the end of the webinar as well. This is a mandatory component of the participation score. Now Eric will launch a test poll so that you have the opportunity to try it out before the presentations begin. So Eric, if you could go ahead and launch that poll.
So the poll should be open, and you can see the question here is what description best fits your position, and you can select one of the following: operator, engineer, administrator, or other. Looks like so far we've got 79% of people voted. We're up to 84. We've got roughly 90% people in right now. Matt, do you know if this is tracking us as well?
It does not track us.
Okay.
I can go ahead and close the poll and we can move on. It looks like the majority of people we have here today are operators, but we do have a range of other people involved as well. Now, in order to go over some of the way to be able to provide credits for this training, this training has been approved by Ohio EPA for 2 wastewater operator contact hours. And in order to provide these contact hours, Ohio EPA requires us to monitor attendance and participation. In order to receive contact hours, you must have the webinar as your active screen for at least 90% of the time during the training event. This means that you cannot use other apps or windows during the webinar. I highly recommend that you close out of anything else outside of the GoToWebinar application, just to make sure that there aren't any issues that come up with not being able to get that attendance score. You'll also need to participate in 3 out of the 4 polls in each presentation. However, your answers will not be graded and accuracy will not count towards your participation score. Immediately following the webinar and before you log out, you'll be asked to take a mandatory survey. The survey counts towards your participation score. Any technology issues such as loss of Wi-Fi will not change these participation requirements, but if you lose your connection, please try to reconnect as soon as possible. I'll also mention that the contact hour request form can be found in the handout section of the GoToWebinar toolbar. Now, today's webinar will include 2 presentations with a 5-minute break in between those presentations. During the break, please do not log out of the webinar, and please keep up the webinar as your primary window, or as the hopefully only window, to make sure that there won't be any issues with the participation score. Now, with those announcements out of the way, I'd like to pass this off. I'd like to introduce to you our first set of speakers. So we have a trio of speakers going over the first presentation that we have. James Boyd is an OEPA Class 3 wastewater operator and serves as operation managers of the Beaver Creek Division for Greene County Sanitary Engineering. And he oversees the operations of 3 water resource reclamation facilities. James holds a Master's of Science in Environmental Safety and Health Management from the University of Findlay, and is currently working on completing his submission for OEPA Class 4 certification. We also have Dr. Susanna Bararova, MPH, who serves as a research specialist in the Department of Civil, Environmental, and Geodetic Engineering at OSU, and the Associate Director of the Ohio Water Resources Center. Her research interest lies in the area of microbial disinfection and recovery in natural and engineered systems. For the past year, she is coordinating the Ohio Coronavirus Wastewater Monitoring Network, a statewide collaboration between university researchers, utilities, commercial partners, and government agencies to protect public health. And finally, we have Judith Strathoff. She is a research assistant engineer at the Ohio State University in the Department of Civil, Environmental, and Geodetic Engineering, an environmental engineering graduate student at Delft University of Technology. She works in the lab to process and quantify SARS-CoV-2 in wastewater and has worked on projects about the impacts of particles on UV disinfection in drinking water, water quality characterization of wastewater from dormitories for SARS-CoV-2, using bacteria to mitigate cracks in concrete, and performing a quantitative microbial risk assessment for using reclaimed water to recharge an aquifer. Now, with those introductions completed, I'd like to pass it on to our presenters for our first presentation of the day. Thank you.
Thank you for the introductions, and this has been tremendous team effort, so it will also be team presentation. So I will start, and then Jim and Judith will present part of the talk. So I'm trying to move the slide. So we'll be talking about the wastewater-based epidemiology or wastewater monitoring for public health. And here is a small, very brief diagram which shows you kind of what it is about, and I will start in the top part of the diagram. So if there is any pathogen or genes from the pathogen present in wastewater because people are shedding it either in feces, urine, or saliva, or any other metabolite, chemical metabolite, you can theoretically measure it in the wastewater from the community. And in that sense, that will be pooled sample because it will represent the whole sewer community. And usually the sample for public health action is taken in the influent to the treatment plant or somewhere in the sewer network. Sometimes it can be also taken in the primary clarifier, but usually it's before any treatment. So that wastewater marker usually is already diluted by stormwater and other industrial flows. So you need to take it kind of as soon as possible. It's coming to the plant. The sample then can be analyzed in the laboratory. In the collection, Jim will talk a little bit more about it. It's analyzed in the laboratory for the marker you want to use. And then it can be used to estimate community prevalence of the disease or usage or any other community health. Usually we recommend, and always it's recommended that it's not used in isolation, that it's complementary to the other public health surveillance, such as hospital admission data, prescription data, and other human biomonitoring. So this is not completely new tool. It's been used, it's been researched for a very long time, over probably 100 years, but really used, applied has been mainly for the poliovirus transmission in developing countries. And World Health Organization have actually even manual for environmental surveillance. So in a lot of developing countries, you don't have centralized wastewater collection systems. So then often you use some type of wastewater collection systems if they are, or even reverse surveillance if they are influenced by the human waste. So it's actually been used to prevent outbreaks of poliovirus. In, for example, one of the most recent use is from actually a developed country, from Israel, where they detected it in wastewater in 2012 and were able to kind of track the disease outbreak and prevent it spreading. And there is also some other international long-term studies of, for example, drug consumption in Europe. And the picture is the map of the European illicit drug use network. They have a public dashboard. And there it's drug surveillance has been used to understand where the public health responses could be targeted. We will mainly focus this presentation on the infectious disease monitoring and not on monitoring of the chemical markers. Other application has been mainly research, and it really became national and international kind of applied science now during the SARS-CoV-2 pandemic. So early in the COVID-19 outbreak, some researchers, especially the ones which already had established the wastewater monitoring of the influent for public health, noticed that the SARS-CoV-2 RNA is present in wastewater. And on the right-hand side, you have a figure which shows you, this is a little bit more recent research, which try to quantify where does the COVID-19 RNA comes from to wastewater, and mainly it's connected with the feces. If you notice, the x-axis is logarithmic scale, so in the feces is 2 orders of magnitude more than in other sources like urine or saliva. So they noticed it's in the wastewater and started tracking it and saw we are tracking it or measuring it in wastewater as an RNA fragment. There is some research going into looking if it's infective, the virus in the wastewater, and it really hasn't been shown. People didn't even manage to isolate infective SARS-CoV-2 virus from the feces. And it gets even more degraded in wastewater. People are looking if the virus is there as a particle or if it's really just the genetic signal, and most of it is genetic signal. There is also studies comparing the wastewater operators or treatment workers with other and looking at the incidence of the disease, and they really don't see difference. So right now the science is showing us that it's not infective anymore when it's in the wastewater. So we're measuring it as just RNA, not as a virus. And then it was shown also pretty early on, and Judith will talk more about that, it's correlated with cases. So the monitoring initiative for SARS-CoV-2 really early on in May, it was government initiative, and we are really one of the first network in the United States, one of the first one to do statewide monitoring, and we are very proud of it. Currently, we are monitoring locations twice a week, and counties and its differences of wastewater treatment plants range from about 1,000 self-residents to 5 million, and at the bottom also that these treatment plants represent. This is the dashboard, which the state is now updating once a week about the levels. You can look at each of these points and what are the levels of the RNA in the wastewater. And I think we got to the poll question.
So this question is, does your utility participate in Ohio's coronavirus wastewater monitoring system? And you can answer yes or no here.
And Susanna, I'd like to mention that there was going to be a little bit of an issue with your connection. So if you want to turn off your camera just to make sure that you're still able to, so that your audio and your presentation will still come through.
Yes, I noticed it, so I switched it off already.
Okay, thank you.
So there's about 90% in now. We'll wait another 10 or so seconds. And it looks like about 60% have answered yes, versus around 20% answered no, and around 20% answered they weren't sure.
That's great. So we will now talk a little bit about the 3 main components of the monitoring aspects. The first one is of course sampling, and Jim will talk much more about it, but generally the utilities collect influent samples twice a week for the Ohio's network, and it needs to be stored in fridge temperature because the virus can degrade in the wastewater, and usually it's sent or picked up by courier service to designated lab. In the lab, and Judith will talk more about it, they're analyzing the sample. We have about 7 analytical laboratories in the state. They have different methods for analysis and about 2 to 3 days turnaround time, some even shorter. And then the data are collected by the Ohio Water Resources Center, compiled and sent to ODH for the displays on the dashboard and trend analysis. So Jim, you can go ahead.
You'll have to unmute yourself, Jim.
Hit one button, forgot the other one. Sorry about that. So my part of this is basically the treatment plant side of things, the collection system, and how we get from the raw influent back to the laboratories. So basically, all of our flows obviously begin at homes and businesses. It comes through our treatment plant, through the collection system. Here we're mostly gravity. We only have about 10 pump stations in the Beaver Creek collection system, so we don't get a lot of destruction of anything, based on pumping or anything like that. Obviously, the time in the sewer can affect the amount of stuff that's present when they get to the lab. Normally, obviously closer, you're going to have higher concentrations. It won't dilute as much, and that's kind of just the normal situation with collection systems. So obviously, the longer time, the more decay that's caused. So basically, we try to make sure that, I know when they were setting up these initial things that some places have much larger systems would actually collect in multiple places in order to get a better representation of the actual levels of the virus. Same thing with temperature and pH. Obviously, that's a known issue, but they overcome that pretty well. So let me go next slide. There we go. So basically, with your sampling, there's 3 basic types of sampling that can be conducted, especially depending on what the need is. You can do a grab sample, which is a quick snapshot, one sample, and off it goes. For analysis, there are systems that do multiple grabs, so you'll actually collect multiple manual samples throughout the day, composite it up, and send it off. And then we have composite-based sampling, which is normally automated, such as those samplers there. So they're all electrically pumped, and it's more conducive to getting a very broad idea of what's going on throughout the day. Next slide. So when it comes to composite sampling, you have a couple of different methods that can happen with. You can set up a time-based system where it's like a set amount of time between samples. We go full proportional. There's a single bottle, multi-bottle setups. I mean, there's a multitude of different ways of collecting samples, and sometimes that's based on your permit. Right now, our permit does mandate that we do flow-based collection. So how we handle that here in Greene County is we have automatic samplers that are refrigerated, so it's all in one unit, and they're on every source. I have 4 treatment plants in the county, and all of them have one on the influent and the effluent, which helps us get some comparative samples based on what's coming in the plant and what goes out. Here at Beaver Creek, because we have primary clarifiers, we also have another sampler after that, so we can actually see how much stuff we're removing in our primaries in order to see what kind of impact that has on our treatment system. We use a single bottle. It's a 10-liter bottle. We pull 100 mil samples. We aim for about 96 a day, which is kind of the equivalent of a 15-minute interval sample, but we are flow-based. So that module there on the right actually connects to our sampler, collects data from our SCADA system, and tells basically based on the number of gallons per interval, and then it'll send the signal to collect the sample. So, and there's a couple of different methods with that. Some places use what they call a pulse system, where the flow meters will actually put out pulses and it measures those. Ours are actually complete flow based, so it actually counts down gallons between the samples. So, next slide. So this kind of outline of how the different types of sampling works. So obviously your time proportional sampling, it's an even time spacing. One of my plants, my class 1 plant, actually doesn't have SCADA for flow monitoring on availability to actually run the samplers. So we actually do time-based there, but we're not required for like proportional sampling or anything. So basically we do that. We just set up that 15-minute interval. It gives us about a full bottle every time we collect a sample. Your flow proportional sampling. So usually, again, it's usually set up to be samples between a set amount of flow that's recorded through your meter. And normally what we'll try to do is we take our average daily flow and we try to multiply that by 90, divide it by 96, so we get a set number of samples. And we have to adjust it based on flows. So if we get a weather event or a higher flow, we need to kind of adjust that so that we're not sampling we get a full representation for the whole day. We don't get cut off early. There is a volume-based one that actually would make the rate between samples shorten based on how high the flow is. I don't know if most places don't use that. I guess there's a logic to it, but I haven't seen anything to make that more efficient. So next slide. So basically, here's your different kind of some of the effects that can happen with the different types of sampling. So obviously with a grab sample, you're going to only get a snapshot from that particular time in place. So you're not really going to get a true representation of what's happened over your full day. So you could have artificially high or low numbers, obviously depending on what your flow coming into the plant is. The higher the flows, if you do a grab, obviously it's going to be more diluted, whereas your low flow times, you're going to actually possibly have higher concentrations. Time-based is better than that, but it still could be artificial because you could be collecting samples at times where you have little to no flow, or you might miss a sample time where you have a higher amount coming in. So the flow and volume base is obviously the best method, and I think that's one of the reasons that most of the bigger plants in the state are required to use this type of sampling for the EPA. Next slide. Yep. So another aspect of the sampling you got to kind of watch for is your stormwater and your concentration dilution. We all know there's two different types of collection systems out there. There's combined systems, which take your sewer flow and add stormwater flow to that, and what they call a closed system, which is sewer flow only. Here in Greene County, we have all closed sewer systems, but that does not really mean that we're impervious from outside influence. So your combined sewers, your stormwater flow could actually tremendously vary your amount coming in. You have to allow for the dilution as far as when you're testing your parameters to kind of understand what that does to your effects, which is one of the reasons why we collect flow numbers from our system in order to let the lab know kind of what we've been experiencing for the day, so they can kind of account for that potential dilution. And then closed sewers, obviously, were designed to transport sewage only. They're not perfect. They're not completely watertight, so we get a lot of inflow and infiltration. That's normal. Another thing that we deal with here is we have some older areas of the county that have sump pumps and gutters and things tied into our system, and it's a political football that no one wants to pick up and carry because it's to cause quite a bit of animosity and extra expense for the customers. So they're trying to put some information out there to educate people, but we're not really going in and enforcing some of those rules that could lower our inflow. So next slide. So here's the other stuff that we get along with our sample every day. So basically we collect our sample, we transport it to our central lab, which is actually on the site here of the Beaver Creek campus, and then they package up the sample for transport to the lab. I know in the beginning we were actually shipping it via UPS, which was not as convenient for us. And I know a little bit over a year ago we started having a courier service, which came directly to the lab to pick up, which helped greatly in our ability to kind of continue on with the program. So we've been happy to do that. So the data we give to the lab, we do the 24-hour flow data. We give temperature readings, so we give min and max pH readings and minimum and maximum temperature readings, and that helps them determine, if things are broken down within the sample. We also run our TSS daily on those lab samples and basically indicate that as well, because obviously the higher concentration of solids, the more of a stronger sample you might have. So particularly at our plant, we sample 3 times per week for our NPDES permit. The COVID sampling actually requires us to do 1 additional sample in order to meet their required intervals. So we actually run an additional sample, and most of the time it's not in correlation with one of our permanent samples. So that does kind of add, it adds numbers that we have to add to our reporting period, just because once you've collected that sample, it becomes part of your system, but so far it hasn't caused any undue problems. So I think that's it.
So the question here is, what type of sampling is primarily conducted at your facility? The choices are single grab, multi-grab, time-based composite, flow-based composite, or volume-based composite. Looks like we got some numbers coming in fairly well here. A lot of folks are doing flow-based composite. Right now we're at 89%, so we'll give it a few more seconds. Okay, 95% are in. That seems to be what we're getting. The results are 44% flow-based composite, and then roughly 20% for each of the rest.
All right, I will take over now. Hi, I'm Judith. I would like to apologize for not having my camera on. Unfortunately, it does not work on my laptop. I work in an environmental engineering lab at Ohio State University, where we process wastewater samples for SARS-CoV-2 monitoring. We receive 1 liter samples twice a week from 5 different wastewater treatment plants in Ohio. Usually these are 24-hour composite samples, and as Jim mentioned, we use a courier service, and therefore we receive the samples approximately 4 to 5 hours after the samples are taken. Before that, we used UPS, and we would receive the samples approximately 28 hours after they were taken. As long as the samples are kept cold, they should have minimal degradation for RNA extraction for 24 to 48 hours after samples are taken. Upon receipt at the lab, we spike 500 microliters of bovine coronavirus vaccine into 1 liter of wastewater, and that's to calculate the recovery efficiency of the filtration and RNA extraction method. We then use membrane filtration to concentrate the solids on the filter. We use a 0.45 micron mixed cellulose ester filter, and we filter at least 100 milliliters of wastewater. And we can sometimes do a little bit more depending just kind of on the flow of the wastewater through the filter. Some labs use a different process to concentrate the solids. They might use centrifugation. We then use the Trizol reagent method to lyse the samples and isolate the RNA. The extracted RNA samples are then stored at -80 degrees Celsius until we perform RT-qPCR to quantify the concentration of the SARS-CoV-2 N1 and N2 gene, and then as well as the bovine coronavirus vaccine and pepper mottle virus, which is a human fecal viral indicator. And I will go into that into more detail a little bit later. Next slide, please. Thank you. As stated previously, to quantify the SARS-CoV-2 virus in wastewater, we use polymerase chain reaction, or PCR. PCR amplifies copies of a specific segment of DNA using primers and probes. It rapidly produces numerous copies, could be thousands to millions to billions of copies, so that the specific DNA segment or gene can be studied in greater detail. PCR involves using primers, short synthetic DNA fragments, to select a segment of the genome to be amplified, and then multiple rounds of DNA synthesis to amplify that segment. The primer is how you choose which gene or which segment to amplify. So we specifically choose primers for both the N1 and the N2 gene. And then probes are fluorescently labeled DNA oligonucleotides. They are designed to bind downstream of one of the primers during the PCR reaction and to give a fluorescent signal during the reaction. Some other methods might use dyes instead of fluorescent, but we use the fluorescent probes. The probe is then cleaved by the polymerase enzyme during the reaction. By cleaving the probe, the reporter and the quencher are separated, which means that the quencher no longer has its effect over the reporter, and the level of fluorescence increases. Based on the fluorescent signal, the quantity or concentration of SARS-CoV-2 can be determined. Digital PCR, or dPCR, measures the absolute copy number directly, and you don't need a quantification standard. You just need a positive control, while quantitative PCR, or qPCR, relies on extrapolation from a standard. Both of them have advantages and disadvantages. We use qPCR, and so we rely on a standard, but then it's based on the quality of the standard. For example, our standard is from IDT, and it's actually double-stranded DNA, while our sample initially starts as RNA. So you have to make a kind of a multiplication factor to correct for that. And then there could also be some issues with the DNA not being linearized, and that could increase or decrease your quantification. Next slide, please. Normalization of SARS-CoV-2 aims to improve the estimate of the population contributing to wastewater. Jim already talked a little bit about this, but variability within a sewer shed impacts the concentration of SARS-CoV-2. This could be due to holidays where people tend to travel a lot, or weekdays versus weekends due to some people leaving for work and they might leave that sewer shed during the day, but not for the weekends, and also weather events such as rain or snow. This figure on the bottom right shows that during COVID-19 restrictions in New York City, the ammonia load, or an indicator for urine, decreased in the Manhattan downtown area and increased in the surrounding areas. This could be due to less people traveling for work, staying home, or moving to areas that are a little bit more affordable, or also that less people are traveling, visiting New York City. And that can have then an impact on the wastewater as less people are contributing to it. The most common markers to normalize for SARS-CoV-2 concentration in wastewater are the population in the sewer shed, flow rate, and human fecal indicators such as pepper mouth mottle virus, PMMOV, or cross-assembly phage, CRAS-phage. Our lab specifically tests for PMMOV. However, some studies have found that normalization does not always improve correlation. And this could be due to having, for example, a years-long longitudinal studies that don't have extreme weather events. And so then just having the variability of doing a study for that long allows for these slight impacts, such as holidays or weekends, weekdays, to be evened out. The recovery efficiency can vary widely from sample to sample, from method to method, and from sewer shed to sewer shed. The method used to concentrate and extract RNA may lead to RNA loss, and it is important to calculate the method recovery efficiency. Low recovery efficiency can lead to underestimation of the actual concentration in the wastewater. Recovery efficiency of a method can be calculated by spiking a surrogate into the sample. The surrogate should be something that is not naturally present in the sample, and that is why we use bovine coronavirus vaccine. And then it must also be something that you know the concentration of initially, so that you know how much was in there when you spiked it into the sample, and then when you quantify the concentration after your method, then you will see how much you were able to actually extract. So then you know the initial and then the final concentration. Recovery efficiencies can vary widely, and that can vary also just sample to sample even, and it can range from 1 to 60 usually, but sometimes it's even 100%. Something that can occur during PCR is inhibition. PCR inhibitors are any factors which prevent amplification. It can lead to underestimation of actual concentration in the sample. It usually affects PCR through interaction with DNA or interference with the DNA polymerase. Diluting the samples could lead to a decrease in inhibition, as less of the inhibitors would be present in the sample. And there's also ways to test for how much inhibition is in your sample. And then some of the kits designed by companies such as Promega, they include steps to remove the inhibitors. And we have our third poll question.
So the question here is, what are the important factors to consider for SARS-CoV-2 wastewater monitoring? Answers range from normalization of SARS-CoV-2, method of recovery efficiency, PCR inhibition, or all of the above. Looks like everyone so far is choosing all of the above. We have roughly 85% in. We'll let it go for a little bit longer here. Okay, we're at 95%. That's what we've been getting. 100% of people chose all of the above.
That's great. So for our next slide, so this image shows trends between cases and SARS-CoV-2 concentration in the wastewater. Each row is a specific wastewater utility location. The red is the amount of cases, while the blue lines are SARS-CoV-2 concentration. For the top row, you can see how the correlation between cases and SARS-CoV-2 gene copies per liter concentration is fairly consistent, whether it's compared to the amount of cases or whether you use the 3-day, 5-day, or 7-day rolling averages of cases. The Spearman's R value remains at 0.79 and 0.78, and the row value does show that the correlation is significant. For the bottom row, this utility showed that using rolling averages actually increased the correlation for Spearman's R. The rolling average can help for some utilities, as in some locations, maybe more rural locations, people might have less availability for early testing if they're feeling sick or showing symptoms, or if they've been exposed to somebody. And so for some locations, some utilities, using the rolling average can help increase the correlation, and while for some other utilities, it won't necessarily increase it. And then this is just another image for one of the wastewater treatment plants in the statewide monitoring project. And you can see how the concentration spikes, where the cases spike. The red is the SARS-CoV-2 concentration, and then the gray line is the cases. Sometimes we see that the concentration in the wastewater, we see that spike before the cases spike, such as right after December 1st, 2021. And that can help with the monitoring, seeing kind of that there's an increase in cases right before people are starting to test. But overall, it follows pretty well. And then in this image, it shows the cases versus SARS-CoV-2 that has been normalized by PMMOV, that's the middle image, and then the crossfage, which is the image on the right. And those are both human fecal indicators. There's an increased correlation between cases and PMMOV normalized N2 concentration, but that change was not significant, just from 0.78 to 0.79. And then there was actually a decreased correlation between cases and crossfage normalized N2 concentration from 0.78 to 0.55. These findings were just for one specific location, but over a very long time period. So some sewer sheds might not need normalization if the study period is over a long time period and it doesn't have those frequent extreme weather events. And then other sewer sheds could have an increased correlation with normalization, which is why it's so important to test for these factors. Then after our lab has quantified the SARS-CoV-2 concentration, we then send our samples to another lab where they sequence RNA. Sequencing is used to determine the entire genetic makeup of a specific organism or cell type. Initially, during the start of the COVID-19 pandemic, sequencing was used to identify SARS-CoV-2, and that was then used to develop diagnostic tests. But now, using continuous sequencing allows for determining new variants present and the proportions of different variants present, as well as lineage of variants. It really allows us to monitor the spread and evolution of the virus, which can be useful for future prediction and responses by the public health. Some variants could have mutations that make them more deadly or allows them to spread quicker than the original virus. The current circulating variants of concern are Delta and Omicron. And then when I checked like the CDC website, I saw many other SARS-CoV-2 variants that have been and are present, but that I haven't heard of before in the media. And that's just because they aren't as concerning as Delta and Omicron right now. And here we can see the proportion of Omicron and Delta over time. The orange is Delta and the pink is Omicron. And you can see how until about December, it was really Delta that was kind of dominating the variant of concern present. And then in December, Omicron really started popping up and became more common than Delta. And I will now hand it back to Susanna.
I only have 2 more slides left, so I just want to talk very briefly about the application. So currently, when we see increases in the wastewater, which are increases for at least 3 samples of 10-fold or 1 log, then the Ohio Department of Health sends alerts to the local health district and also to the utility. and they usually interpreting the results, you know, with the knowledge of their community and their wastewater systems. They are also provided a communication toolkit and then access if they want to tracing, now mostly vaccination and state vaccination and testing teams. We have an online dashboard, which I showed snapshot, which is currently updated only once a week. And then we also participate in the CDC National Wastewater Surveillance System, and there is a snapshot of their dashboard on the right here. And then last one is the future of the wastewater monitoring for public health, at least in Ohio. So it really, the interest right now, it's on a voluntary basis, and it should be public health and community driven. But we are already looking, or the whole United States and world is looking at other markers in the wastewater, for example, seasonal influenza and foodborne illness, which both been shown different foodborne illness bacteria and viruses. These all can be monitored in the wastewater and could help kind of the public health, mainly for targeting resources, for example, in the state. So this is something which the CDC is developing methods for and will probably start being implemented over the next year. And I think that's all.
So the last question has to do with the interest of your monitoring for your community, and what we're looking at, choose all of the ones that apply, from seasonal influenza, antimicrobial resistance, foodborne outbreaks, illicit drugs, or other. Okay. We got about 85% in, 40 seconds. So we'll go another 10 seconds, try to get 95%. And okay, everyone's voted. Looks like the majority are showing for seasonal influenza, 50% for antimicrobial resistance, 39 foodborne outbreaks, 33 illicit drug use, and 22% for other.
So that's the whole presentation. So are there any questions? I want to open it for questions.
Yep, I have a question for Jim, and I'd also like to mention for anyone on the webinar that you can use the question function through GoToWebinar to put in a question to our presenters, and a TMACOG staff member will read it out. What I was curious about, so this one's for Jim, how easy or hard was it to implement this testing regime into your daily, your routine operations that you're used to? Was it hard to be able to have that additional time constraints, or how did you see that as being challenging or not to be able to add that into your routine?
Yeah, it wasn't extremely difficult. The only thing that changed a little bit is when they started the courier service. So we arrive at 7 a.m. We're a single shift operation. So it kind of forced us to get our sample over to the lab a lot quicker in order to have it ready for the courier to arrive. And then the extra sample, like I talked about, that's kind of a little bit of annoyance, but we already run our effluent sampler daily anyway and do in-house like process control testing with it. So it wasn't terrible, but it did take a little bit of adjustment, and it's a little bit of griping from the guys, but they recovered well and they just deal with it now.
Okay, so there's a question put in here, and it's how long are we going to continue COVID wastewater testing?
I think that the CDC is funding another year of COVID wastewater testing. I think it will really depend on where this pandemic, it's not pandemic anymore, like how this disease is evolving and developing. Right now that we don't have much information about clinical testing because people usually get tested with the at-home kits and don't report the disease. So the wastewater is really becoming the primary tool to just see how it's progressing in the community. So I don't have exact answer, I'm sorry, but for sure 1 more year.
Thank you. I'm not seeing any more questions in here. If anyone wants to go ahead and add, by all means, go ahead and type a question in the box here.
No, I have a question. In general, were there challenges to getting kind of this system up and running, or developing the methodology to be able to get this in place kind of relatively quickly, considering how things moved along?
There were huge challenges because it wasn't, the sampling for the influent wasn't developed and the methods weren't developed as well. There was a lot of different methods, and even now our labs are using different methods. There is not one standard method. And we developed the network in basically 4 weeks. So there was a lot of challenges and really took a lot of people which worked on this to have it deployed. So I don't know if you have any specific, we are still learning. I think the same way we are learning about the disease from a clinical perspective, we are still learning about the wastewater monitoring as well.
So we have another question added in here. This is for any of the speakers. What have been your most unexpected aspects of this process? Was there anything surprising or unpredicted?
I guess I can answer that one. I didn't start working with, I mostly worked on drinking water, and I didn't start working with wastewater until I was doing the SARS-CoV-2 monitoring, whether it was on campus and then later with the wastewater utilities. And I was pretty surprised how, I guess, I don't want to say clean, but not like how it didn't look as dirty as I thought it would. I thought the wastewater would look a lot grosser. And even though I collected them by the sewers on campus, so pretty close to the source, I would say it was a lot. I guess, people use a lot of water for showering and washing their hands, so not as dirty as I thought it would be.
I mean, there was, I think, a lot of things I did not expect. So Jim talked about the delivery, and I discovered how difficult actually it is to get same-day delivery. We are trying to look at it for all the sites. We have 70 sites, a little bit over in the state twice a week, and it's just logistical, very difficult logistically. So I think we first time thought that shipment will be okay, but because we want the results as soon as possible, and because surprisingly shipping is really not reliable, we had a lot of samples getting lost. So we moved to the delivery. So I think that's my kind of big surprise.
Yeah, I think for us, it was just seeing the dashboard and tracking the data and how really the spikes on the testing shows up about 5 to 7 days before your case numbers went up. So it was kind of neat to see the correlation and actually how we're able to kind of predict some stuff that's going to happen. So that was nice for us.
Looks like there's one more question here having to do with what's the cost of testing a sample for COVID?
So right now the utilities are doing it voluntarily, and they don't pay anything. I think the cost per sample differ largely because it's still not standard anything. Judy, do you have estimate per sample how much it? cost?
We did calculate it just based for our lab specifically. And I'm trying to remember the cost. It's fairly expensive.
But just to put it in comparison, so we are basically testing, thousands to 500,000 people and the trend in the community. The cost is comparable to cost per, of the PCR cost of person. It's very similar method. It's the same method. The ones you extract, the extracting of the RNA, the concentration is not same, but that's the only unique part of the wastewater. So that cost is comparable to the clinical test of one person.
I don't see any more questions in here, so looks like we're pretty good as far as that goes.
I know I have another question. I guess one of my concerns would be, especially with this variation in methodology, do you see that changing to really come back and have something more unified? And that kind of goes for Jim as well on the wastewater side of, is it the same methodology for the collection? You'd mentioned that there are different types, but kind of, do you see more standardization happening as this becomes, I guess what I'd say, more routine or, more mainstream?
I do know a couple of manufacturers are trying to lobby for to get more samplers out in the field to try to make that happen. There's some programs out there to help actually get some of these refrigerated, automatic samplers put in place, especially for, communities that are smaller that don't have the requirement, built into their permitting. I think if they can get that accomplished, it would definitely help on the standardization on that side.
For the methods, we are not comparing kind of between sites, but only looking at the trend at single sites. I don't think the US EPA, I don't think they are preparing standardization of the method, but I know that CDC is starting to prescribe some things they would like to see, like using digital PCR, not qPCR. And also we are moving in Ohio to single lab, to the public health lab, all the sites starting July. So that will assure that there is a one method, which is pretty consistent with the other public health departments. So there is push for some consistency, but I don't think there will be a single standard method for the SARS-CoV-2.
Another question I'd have is, where do you see kind of this going into the future? I know we had talked about some of the other types of things you could test for, but is this something that you had kind of seen as a focus previously before COVID? And do you think this really will provide that incentive to be able to continue doing this type of work and expanding it?
I think it wasn't widely known before COVID, but it really became kind of on the international front of the response to SARS-CoV-2, and it really kind of did that huge jump from research to application during the pandemic, and I think it's going to stay in the application. I'm quite, even if you talk internationally about the wastewater monitoring, it's being now applied widely, and I think it's going to stay here as one of the public health monitoring tool.
I have another question for Jim. When you're saying how it's this monitoring is kind of built into your permits, Can you explain that process and kind of how that was built in and why it's different between larger plants and smaller ones? Can you kind of describe the process of how you were involved and how that works?
Yeah, so basically our MPDES permit, which is issued by the state for us to operate and discharge to the waterways, basically outlines your frequencies, your methods, things like that. So what happens here is that my big plant here is a class 4 plant. We're required to test 3 days a week. I have a class 2 plant that only has to test twice a week, and my class 1 only has to go once. So that frequency is kind of built in. They don't dictate which days. They just have to be representative of your flow. So, we do it on a schedule that kind of works with our lab staff so that they don't have to come in on weekends and do, read results and things like that. We do take effluent samples at all of our plants every day, like I said, for process sampling. But as far as like the standardization and stuff, it pretty much comes from the EPA and whatever's written in your permit. So, we're kind of told what we need to do, and then anything else that we do is for ourselves. I will say though, if I ever send extra samples over to the lab for them to record data, and it is a parameter that's in my permit, that has to be reported whether it's needed or not. Good example of that is I actually test phosphorus weekly. We're only supposed to do it once a week, and now we're actually testing every sample that goes to the lab to kind of help get a better picture of how well we're removing that from our effluent. So, but the downside is, while I can get a better idea of what we're doing, I also have to report all those numbers. So.
There's another question in the box here. Have you had any municipalities decide to no longer participate in the testing program?
Yes, we had really few, but we had municipalities which stopped participating. As I said, participating is voluntarily. So if they decide they don't want to participate, we try to persuade them, but they can withdraw whenever they want. So I think we have 3 or 4 which stop participating.
Did they provide any reasoning, or was it they just told you, no, we don't want to do it anymore?
We usually ask, so I need to know, last time it happened was a long time ago, but so oftentimes has to do with staff shortages. So there is some burden. Of course, we are trying to, and I didn't realize, Jim, and I need to talk about it, about the monitoring to higher EPA once you take samples. So we are kind of trying to minimize the burden as much as we can, but we are not always knowledgeable on the, exactly what everything you have to do. So it's a lot to do with staff and smaller utilities, for sure.
I've got another question for Jim. Do you see any other types of projects that you'd be interested in doing collaboration, or do you kind of see a little bit, some of this COVID monitoring being somewhat of a one-off type, project, since it has been, so big? If you're keeping with, would you be keeping with some of the wastewater monitoring in general? And then are there any other types of projects for wastewater monitoring that you're potentially interested in?
Yeah, I know that when the talk was out there, whether we could actually drop out when that time period kind of came around, it was really conversations between us and our public health director for the county. And they're the ones that pretty much pushed us to stay active because they really value the data. So I'm assuming if they are able to develop and get some other beneficial type testing, that it'll probably continue. Obviously, that is going to be kind of dictated by the cost. If it ever came to the point where we're going to have to start, paying and have an annual, outlaying of funds, that could totally change the way things go. But as far as long as it's covered and it's really no major hassle, I don't see why we wouldn't do it to try to help. Just keep an eye on some of those other things like they talked about with the different types of infections, diseases, and things. And I mean, I don't know that I would best necessarily continue it for just tracking drug use and that kind of thing. To me, that's a little bit of an overreach. But anything that could definitely affect the overall public health with infection rates, it's probably a benefit, in my opinion.
And kind of with a follow-up on that, are there other types of things, outside of just testing? Are there any other things that you've heard about for, how wastewater plants can also be tied in with local health departments?
I mean, there's been no concrete talks of anything. I know some of the stuff I've heard, they're really looking for those seasonal type diseases that can really, really affect everybody quickly, your flus and that kind of thing. But I haven't heard of anything coming down officially as of yet.
All right, well, we're about at the time for the end of this presentation. I'd like to thank everyone for presenting on this incredibly interesting and timely topic. I think it's something that has really been new and kind of exciting to be able to get these partnerships between wastewater and health departments and really show the collaborative nature of how we can deal with these issues as a society. Now we'll be moving on to a 5-minute break. I would also like to note to everyone that they should keep open the GoToWebinar, just so that, again, they're not having any issues with the participation scores. And also I'd like to note that the operator handout, the operator contact hour request form can also be found in the handout section. So I'd like to, with that, we'll take a 5-minute break and return at 1:14. Thank you. All right, after that 5-minute break, I'd like to start with our second presentation of the day. Dan Miller received his BSCE from the University of Toledo and is a registered PE in Ohio and Michigan. Currently, he is working as the Director of Wastewater, Design or Project Manager on wastewater projects at Jones & Henry Engineers. Mr. Miller has spent 39 years in the wastewater industry, 35 years at Jones & Henry, and 4 years at Pelton Environmental Products. He has presented previously at the WF National Conference and state WEF conferences in Ohio, Michigan, and Indiana. He is also the winner of an Indiana ACEC Honor Award for the Class A Biosolids project at Carmel, Indiana, and an Ohio ACEC Honor Award for the dewatering work in North Baltimore, Ohio. And with that, I'd like to hand it over to Dan for his presentation. Thank you.
Thanks, Matt. Just kind of checking in. Can you hear me?
Yep, we can hear you and see your slide.
Okay, great, perfect. So we're going to talk about dewatering for small plants. I want to thank Matt for that introduction and the opportunity to be a panelist. And like I said, I want to talk about dewatering for small plants. As consultants, we have seen a lot of more smaller plants ask for dewatering based on lots of different needs and what they're seeing out there as far as land application and land filling. So that's basically what the presentation is going to be about. We'll go through some ways to do dewatering for smaller plants. We'll start with the simplest and then we'll get into some mechanical needs. Since most of the audience is operators, you guys know there's two wastewater streams that we kind of look at. There's the wet stream and the solid stream. The wet stream is basically, you know, the focus of your NPDES permit, and then the solid stream on whether you're digesting some form or other, and then the disposal side. So that disposal side is the one that's kind of getting tricky nowadays through the new regulations and everything else. And kind of dovetailing into the last presentation, how scary is it for maybe operators or everyone in the future that we can measure such things like viruses in wastewater when we start to talk about microconstituents and what other things can we measure in wastewater and what may we have to take out of wastewater in the future. But that's down the road, so, but very interesting presentation prior. So now I'm just trying to move to the next slide. And come on, move. All right, there we go. So in Ohio, what do we call a small plant? You know, as far as minor plants, I pulled this off of the Ohio EPA's website, and I hope I, you know, extrapolated from the Excel sheet. But there's 1,976 minor plants in Ohio, meaning less than 1 MGD. And this doesn't count the 1771 semi-public plants. Now, those are very tiny plants. It might be a isolated school or an isolated industry or a mobile home park that are very much separated from the municipal side. So, and I'm really not going to talk about those type of facilities for the most part. Keep in mind that some of those minor plants are lagoon plants, so they're not a mechanical plant, so they don't have to worry about sludge disposal, so to speak. A 1 MGD plant, you have a population base around 5 to 10,000 in that neighborhood. When I think of a small plant, I would probably include it up to about a 2 MGD, but a good minor category, a good cutoff would be 1 MGD. Just kind of in our neighborhood in the Northwest District, I think when I look, there are 475 total plants. On the minor side, on the municipal, there were 64 that were less than a 0.1 MGD. There were 53 between 0.1 and 0.5 MGD. And then there was 35 between 1 and 10 MGD. So I don't have a breakdown how many ones were there or just near 1, but that's the breakdown that was there. If you look at the picture here, there's this is Ohio City. It's an aerial view of the plant in Ohio City, just kind of west of Paulding. So in their plant, they have an oxidation ditch and 2 final clarifiers, aerobic digestion, and their final disposal method over here, they have a liquid disposal, a liquid tower that they can dispose of, and they also have a couple sand beds. And during this particular project, we designed for them just a simple sludge storage area. All this is paved pavement and highway barriers around the outside here. So very simple for them to take their sand bed dry solids and put them over there for extra storage. So a lot of the original design parameters for a lot of plants went and used liquid as their disposal method for land application. It was pretty easy to store in a tank, put it in a tanker, and then off to the field jimmies for disposal for a lot of plants to be utilized for a long period of time. And then many smaller plants also had sand beds, as you saw Ohio City, as well for disposal. So, you know, over the years, that was really easy. What kind of happened along the way? Well, along the way, that land application periods got to be a lot shorter, and there is an extreme concentration of time and plant personnel to dispose of the liquid solids in a very short period of time before the farmers needed to get on the field for the most part. And then with the staff reductions, that even became shorter. Also, there's a growth in liquid disposal or companies that would dispose of your solids for you. I think that's actually declined here recently. So they kind of took over the liquid disposal side of it. As far as sand beds go, I think as design engineers, we were probably a little overly ambitious on how quickly the sludge would dry out and make it kind of a usable to land apply it. If it's too wet and you try to put it in a spreader, it'll just kind of clump up and plug up the spreader. And so, you know, it had to get fairly dry to come off the spreader well. So that takes longer in the sand bed. So really the sand beds were just most of the time kind of small. So as people actually moved to liquid storage instead of their sand beds, and then they kind of converted the sand beds over most of the time for some kind of storage around the plant. They're also really useful for, you know, putting your grid or sewer cleanings on a sand bed to help along that line. So then finally, sand beds were pretty labor intensive. You know, they're you know, as staffs got smaller, you know, it was difficult for plants just to keep up with the amount of sludge that they were producing. One other thing, and I'll kind of mention a little bit later on too, is that on the liquid side, you know, our wet stream, we're always trying to do better on the wet stream. Well, every time we do better on the wet stream, we're going to make more solids. So that kind of led to, you know, needing more different ways to get rid of your solids. So, you know, why do some of these plants need dewatering now, other than, you know, hey, we're running out of staff? Kind of look at the 3 buzzwords we always see: flexibility, sustainability, and economics. I hate to use the buzzwords, but in this case, they do apply, and I'm going to cover each one of these. And we've put a lot of small dewatering mechanical systems in the smaller plants lately, and they are making very good use of them. So we'll talk about each area here real quick. On the flexibility side, you know, that's kind of the chief thing we've seen really that's driving it, flexibility and economics. You know, land application has always kind of been the way, and land application, due to some new regulations, has gotten tougher for communities, and the periods also seem to be getting shorter now. Whether you want to knock that off to climate change, but we've had some wet times here lately, and if you're putting on fields that farmers are going to use and they got to get in on it, they want you to get out right away in order for them to do whatever planning they have. So we've also eliminated the, you know, applying onto frozen ground, so that's kind of shortened the time period as well. So land application periods have gotten shorter, it seems like. You only have really a few times, you know, in the spring and some when you get the weed off to get your stuff spread out there. As far as regulatory changes, certain the phosphorus changes have increased the land use, or maybe has increased the driving distance you have to go to land apply your sludge. And certainly as soon as you do increase the distance, you're going to increase your costs. You know, part of that too is better farming practices. You know, the farmers are recognizing and they don't want to put too much phosphorus on their crops, or too much fertilizer or nitrogen or nutrients. And they're recognizing too that they're running off if they're overdone and going to, you know, Lake Erie, possibly in our area. So, you know, they're trying to minimize the fertilizer application rates. Environmental groups, you know, they're always kind of out there spreading human waste or spreading sludge on the land. You don't hear too much of it, but if you happen to Google sludge spreading and land application, you'll come up with some pretty interesting stuff that's very much against it. You know, why else are people looking to go to land application or to make a cake? One thing is the landfills are always open. They're not open on weekends necessarily, or holidays, but weather's not an issue. So if you have a really wet period and you can't get your cake onto land app, and let's keep that in mind, that even though you're making a cake, you can still land apply it, or you can take it to the landfill. So there's your big chief flexibility item. So you can always take that material to the landfill if you get in a bind. And then as far as emergency storage, if you have a bulk liquid storage tank and you can't get out on the fields, you quickly cannot build another liquid storage tank. You either have to get someone to take your liquid and store it in there, or you're going to have to call in somebody to process your liquid out with a portable belt press or something like that. As far as cake storage, if your cake storage is full, it's a lot easier to put together some temporary cake storage in a paved area that has a drain to a plant sewer and some highway barriers very quickly to emergency store some cake, or you can emergency store it, on your old sand drying bed. So just to, you know, as an emergency aspect, you can store cake much easier that way. As start as sustainability goes, you've seen more difficult regulations on land application, and they've gotten more restrictive, and quite frankly, regulations normally don't go in the opposite direction. They'll probably get more restrictive in the future, which will make land application even harder for Class B product. May drive some of you guys to a Class A biosolids, but then you got to figure out where you're going to get rid of those solids as well. So it's a balancing act, certainly. Phosphorus obviously has made it more restrictive. you know, we got the PFAS and the PFOs out there now, and that's really thrown a monkey wrench into some of the communities. Michigan has put out some regulations which are kind of middle of the road regulations if you have PFAS or PFOs in your sludge and land applications. So they've kind of set a middle bar as far as what the concentrations can be in their sludge. I understand that Maine has taken the approach that, hey, you can't put any materials, any PFAS or PFOs in your land app. So if you have it, you're stuck. You're gonna end up going to a landfill. That really makes it difficult. So again, that third item there, as far as sustainability, it gives you a second option. You can still land apply cake, and so you can utilize both liquid and dry cake application. What's the future hold? As we heard earlier, we can measure RNA from viruses. What's the next chemical that we're going to find? PFAS, PFOs. You know, there's stuff that we probably don't know about that's out there right now. You know, microplastics certainly are out there that are starting to get measured quite a bit here lately. So will there be an avoidance of putting microplastics out on the field? So who knows? But again, we're kind of looking for some sustainability options here. And then far as economics, you know, lower limits. And when I talk about lower limits, I'm really talking about lower limits on your wet stream process. More sludge will make more sludge, for example. And certainly phosphorus removal is creating more sludge, especially if we're using chemicals to get rid of the phosphorus, like a lot of communities are doing. So it's not a big bump, but it is a bump in your solids production, maybe 5% more solids. I always remember the superintendent down in Ada, Ohio, would say, every time you guys do an improvement here, I make more sludge. And that's true. We're trying to make the wastewater cleaner, and the way to do that is get some of the solids out of it. So reducing your TSS, reducing your BOD and ammonia, you're going to end up making more solids unless you have a really, really more effective digestion system. Some of the economics, again, cake storage is less expensive to build than liquid storage. If you need more cake storage or you need more liquid storage is getting to be pretty expensive. Tank costs range, depending whether you're a steel storage tank or a concrete tank, I'd say 2 to 4 cents a gallon. Where if I'm going to store cake, I can lay down some pavement and put in, again, some highway barriers and make sure that pavement sloped to a drain that heads to the plant. But I can build quite a bit of cake storage for a lot less than we can build liquid storage to get you through, you know, the winter time or cake application periods. There's less paperwork when you have to take things to the landfill, so less manpower, less time. Certainly if you're still applying liquid yourself with a field jimmy and a nurse truck, you know, there's going to be a lot less time. Like it says there, if you're land applying, I think a lot of communities have gotten rid of their own land application programs and are farming out, but there's still some of you out there. There's less risk if you take your cake off to the landfill. You know, what's in my sludge? How am I affecting my neighbors? You know, all those things come into play as far as it is your sludge, so to speak. And if I take it to the landfill, it's kind of more, I don't want to say it's out of sight, out of mind, but now it's at the landfill and I don't have to worry about, you know, maybe what's in my sludge, it's already going into the landfill. As far as some of the economics on the dollars and cents side, and I don't know if I've updated these numbers here just recently, but on the liquid disposal, if you look at 5 cents a gallon and 2.5% sludge, and that's probably aerobically digested sludge, I'd put things in terms of dollars per dry ton, and you're looking at $479 a dry ton. Taking the same landfill disposal at $50 a wet ton. At 18% cake, it works out to about $277 a dry ton. Now, keep in mind that number doesn't include what it costs to make that dry cake. That, you know, whether you're pressing it somehow, when that generally runs around about $100 a dry ton. So I'm going to open it up to poll question number 1.
Okay, which of the following is not a reason small plants need dewatering? Flexibility, economics, odor, sustainability, or none of the above? We're about 75% in at this point. We'll keep it open for a bit. Almost 90%. Okay, 100% are in. We got 6% answered economics, 33 odors, 6% sustainability, and 56% none of the above.
All right, so let's talk about, you know, what are your dewatering options for a small plant. And I'm going to start with the simplest and work our way up to mechanical. And they're all options are on the table, so to speak. So start taking a look at them. The simplest would be a sand drying bed. A lot of sand drying beds were put in many years ago. You just simply dump the sludge on it and let the sludge dry out itself through the weather and the sunshine. They're very simple to operate. They are kind of labor intensive. Again, you're going to have to get in there and take that sludge off and put it in some kind of disposal vehicle, a truck or a dumpster. You're also going to pull up some of the sand on the bed. Over the years, a lot of communities and guys that use sand drying beds have made a lot of design improvements to them. They've improved the runners that they put on the beds, the concrete runners. Usually what you see is they put in the runners based on the equipment they have to take the sludge off the bed. They've added multiple feed points. That's helpful. Instead of just trying to slug it all down from one end, multiple feed points allows the sludge to spread out more evenly across the bed. Some plants have used polymer with mixed success. Again, we talk a little bit about the labor and equipment, you know, to clean the beds. And if you're not using your beds for your sludge, you can always use them for sewer cleaning and grease and watering your grit as well. There's a typical cross section here, sludge sitting on the top. Usually people run it about 8 to 12 inches, their sludge depth when they pour a bed. You have another 12 inches or so of sand, and then another 12 inches or 18 inches of gravel down here, and then some form of usually about a 6-inch pipe to carry away the filtrate that runs through the sand and the gravel. When you go to pull your sludge off, you'll usually disturb part of this upper sand bed, and you have to replace that sand periodically. But they're tried and true. They've been around for a long time. If you ask me, oh, do you guys ever put in sand beds anymore? Yes, we do. We have just put a few in recently. So they're not completely out of the market, but normally if they're going to go to a sand bed, we'll see a client request more of a mechanical dewatering device. Now, there are some improvements made to sand beds, and they're called these quick dry beds. There's a couple of them out there. One that they put sand on the top, and they put this quick dry drainage material, kind of looks like an egg crate, right in here. And inside there is some pea gravel. They add polymer to the mix, and then they put their outlet right in the middle. And you guys already know polymer is a great flocculating agent. And so as soon as you mix polymer with the sludge, it'll start to flocculate the sludge almost immediately. And the water will separate very quickly. So the idea is to try to get the water separated from the sludge a lot faster, and then to get the water to move through the sand and that quick drying zone bed. And I'm just going to kind of jump real quick to the next slide, because I've seen more of this next type than with the sand, where they actually use a plastic piece of media. And this is a wedge wire style media that sits on top of this style of bed. They're pretty effective. You can't pour them as thick as a normal sand drying bed, but you can get them to, with the polymer and the water rushing away through the pores or the wedge wire in these beds, you can get it to dry out pretty quickly. And be removable, I would say, in about 2 to 3 weeks. That's without any rain if we're lucky. So the natural tendency sometime is I'll just cover them. Well, we want the sunshine too. So you want the sunshine, and you want as much dry wind to get to it as well. As the farmers know, a little bit of sunshine and some wind will dry stuff up real quickly. So you want both those elements in there. You can load them at a higher loading rate than you can a normal sand drying bed. One thing you have to do remember though, as you start to return or pour a quicker bed, you're also going to return higher loads to your wastewater treatment plant. So you got to be a little careful in that regard, as well as you don't want to overload your plant with high ammonia loadings, for example, and cause you a violation. So there's limits to how fast you can load some things. Here's a dumpster style system. I can't say that I know of anyone using a dumpster style system. I would say it's really for a pretty small plant. It's a completely contained dumpster, and there's wedge wire on the side of these dumpsters, and you could pour multiple loads in. Again, you're going to use polymer so that you can get the water to flocculate and get out very quickly. Eventually, you'll need to clean the inside of this thing with probably a high pressure hose. I don't know if you actually have to get inside. This particular one has a cover on it, so that's fairly interesting. I'd probably think it's going to be the cover's there to help in transportation of the sludge. You could set it on your drying beds. And then the container, actually, I understand they can be made to match what your hauler container is going to be. So here's some drains here on the side. So as the water, the filtrate drains out, it can be piped to wherever you want to pipe it, or maybe you're setting on a drying bed, it can just go out to the drying bed. Again, I haven't seen too many of these. What I've seen a lot more of, and you guys are probably much more familiar with, are Geo Bags. There's been a lot of Geo Bags in use. This particular one's sitting in a dumpster and placed on somebody's drying bed or somewhere for the water to leak out of the dumpster. But A Geo Bag's a polyethylene bag with small openings in it, filter size openings that basically, when you mix the sludge with the polymer, that you're going to get this water to separate. We'll call it the filtrate to separate. It'll pour out through the openings in the Geo Bag and into that dumpster, and the dumpster then has drains, and the sludge will stay inside the bag. In this case, there's no need to open up that bag. You can use this style of system multiple times. The bag will end up, and everything will end up going to the landfill. So a couple things here again, and then you can match the container with the hauler. A couple things to certainly remember, that you're going to have a polymer feeder and you're going to have some type of feed pump here, and you need to make sure you're matching the rate of your feed pump and your polymer feeder, so that they can feed the correct amount of polymer with the sludge flow that you're taking to your Geo Bag. So I believe I got another poll question coming up now.
Okay, in this one, you're going to select one of the oldest forms of dewatering from mechanical dewatering: sand beds, Geo Bags, quick beds, or all of the above. We're about 90% now, a lot faster on this question, so we'll just let it go a little longer. Okay, 100% are in. We've got 83% answered sand beds, and then 6% for the following: mechanical beds, Geo Bags, and all of the above.
So this is probably what I've seen for a lot of smaller plants more often than anything else, is a kind of a repurposing of their sand drying beds using a Geo Bag. And again, they place a bag right on your sand drying bed, mix the sludge with some polymer, and you can use the bag over and over again. Not the same bag, but you can feed it multiple times. Eventually, you'll need to break that bag open and unload it. And that's probably what I've heard to be the biggest drawback. And, oh, I've got beat the bag open. So, well, you're going to break it open and unload it. It can be a pain. You can think of, I got a 3-foot bag of gelatinous kind of sludge in there that I now got to load up into trucks and get rid of. So again, it can be time consuming. You're going to need equipment, obviously an end loader, but a lot of plants have gone to Geo Bags. I know they use them in Delta, Ohio. They use Geo Bags sitting right on their sand drying bed. So I would, again, I guess to me, I think Defiance also uses some Geo Bags once in a while. To me, the biggest drawback is the breaking of the bag and unloading it, and the time it takes to do that, and probably more likely the mess it takes to do that. I got a couple more here. I've seen this. This is from Blue River Technologies. I saw one of these over in Indiana. I can't say I remember exactly where. I didn't write it down, but it's a vertical Geo Bag with some Bombay doors that you can see there, and a system where so when they fill the bag here from the top, the water runs out the side of the Geo Bag, runs down the sides of the Geo Bag and off the tarp that's sitting on this dumpster. And then once the dewatering kind of operation is complete, you take off the tarp and you open up the Bombay doors there to unload the sludge. So One thing it does allow you to reuse the bag, maybe a few times, but I would think eventually your bag is going to get clogged with some sludge particles and going to be less effective probably every time you use it. Certainly there's a kind of a reduction in the labor cost here in this style of operation because you're dumping it straight into a container. Notice really nice here, this container is lined. I'm sure anybody that takes containers of sludge, they line their containers so that you're not dripping material along the roadway. So just pointing that out as a design feature to anybody here that might use it. So again, Geo Bags really popular in Ohio and Indiana. They're really simple to set up. You know, I would say, hey, you don't even need an engineer to set them up. You can do this all yourself. Order the bag, set your piping up, get a polymer feeder. You don't even need a contractor or anything else like this. So a lot of plants set them up. I know, at least when I did some research before, there was at least 100 systems in Indiana, and I guess the number is probably pretty similar in Ohio. So Geo Bags are really, really popular ways to go for a quick way to get some more cake storage or get more production out of your sand drying beds. Somebody sent me this one. I'd never seen them. In the round, someone pointed it out. So again, you mix the sludge in the water, and this thing turns at a very slow rate. You know, and you can put it on a truck and drive it around. So I would think it might have an application where you're a county or a community that's taken care of multiple really small areas that have some liquid, that store liquid sludge. And that way you can go around and drain out some of these liquid storage facilities, small plants, put them in the round, and then end up taking this and dumping it out, you know, somewhere at your own facility. But again, I can't, someone sent it to me. I've never seen one in use. So, but I thought I'd put it out there. So those are probably the simplest ways to accomplish it. Kind of what's your next step? And the next step is mechanical dewatering. If you don't like what you saw any of those there, or the size of your plant dictates that you're gonna want to look at mechanical dewatering. Some of the reasons why the mechanical dewatering is pretty easy. I would say it's less labor intensive than a Geo Bag or some of those other facilities. Gives you a lot of flexibility. You can run your system anytime you want and haul to the landfill to get rid of sludge. I think really what you got to kind of figure out is what's important to your plant. Labor, is that a big issue? Economics, you know, sustainability. Do we think that we're going to continue to be able to haul liquid at a cost that's reasonable? Or do we just want to start to make a cake and have the option to take the cake to landfill or take the cake to land apply it? So gives you that flexibility and possibly lower the cost. As far as, how am I doing time-wise? 153. As far as mechanical dewatering, you know, it's probably not as expensive as you think, especially if you have a building already built or a facility that you could put a small mechanical facility in. There's lots of different options. Belt presses have been around a long time, either single or dual belts. When I think of a single belt, I think of a Ortec unit. There's a few of those out there. A centrifuge is, you know, a more high-speed device that'll get you a dryer cake for the most part. And then there's screw presses and volute presses. All the manufacturers have scaled down. That's really important. You don't have to get a big dewatering device. They make smaller dewatering devices. Thus, some of the costs have gone. They're easy to operate. They're not difficult to operate. And you can also get them on a skid unit. So I believe most of the manufacturers make a skid unit that'll include the dewatering device, it'll include the feed pump, it'll include the polymer system, and a conveyor to take to discharge the sludge away from the unit. So kind of simplifies things. I guess when I think of a skid unit, and in this one, and here this one was a skid, you can see there's the polymer unit and this small belt press here, and then the feed pump is tucked underneath here. So things start to get compact. So it means that maintenance might get a little more difficult. You can get to the stuff, but it's just a little harder to get to when you compact everything on a small skid style unit. So, what are your design decisions? The first is your device type. What kind of device do I want to use? That's a real critical decision. And there's a lot of choices. And I'm going to kind of run through some of the choices here in a minute. How often do I want to run? That's really a determining factor as far as the size of the unit you need to. And the other big determining factor as far as the size of the unit is, what do I have upstream? How long of storage or how much digestion storage do I have? If you have a lot of digestion storage, you may decide that you don't need to run the unit very often. 1 day a week, maybe, 1 day every 2 weeks. That really enhances some of your opportunities as far as the size of the unit. How do you store your product in the end? What's your disposal method? And what I mean there is, are you going to go exclusively to the landfill, or are you going to go land application? So if you're going to go exclusively to landfill, water costs money at the landfill. So you're going to want to try to get the driest cake possible as far as your disposal and disposal options. As far as disposal options, am I going to truck it? Am I going to take it into dumpsters? Do I have equipment around here? So it all goes into, how am I going to set this up? What are my design decisions? You don't necessarily need a truck to haul it anywhere or end loader. You could set the equipment up so that it dumps right into a dumpster. You set it up high and it discharges straight into a dumpster. So kind of talk about some of the options here. Hope I didn't pass a poll question. I don't think so yet, but I may. I don't think I did. Did I? No, I don't think I did.
I didn't see it.
Here's a belt filter press. Hey, I didn't put, there's not a name on this one. Belt filter press is a real simple device. They're around for a long time. When I started, that was kind of the only dewatering device out there, mechanical device that was, fairly reliable. There were centrifuges out there. They seem to be a bit more unreliable than a belt filter press. So it's a really big workhorse in the industry. Kind of why have communities gone away from a belt filter press? One of the things is a footprint. It's a fairly large footprint. It also uses a lot of wash water. So a half meter or a 1 meter belt press will use 50 gallons a minute of wash water, which is a load coming back to your plant as well. It's an open design. I think, to me, an open design has a lot of pluses. You can see what's going on, but with an open design, and I know they make some of these units now enclosed, that there's kind of an aerosoling going on because you have belt washes and kind of a lot of moisture around a belt filter press operation. So just kind of running through how it works. So sludge comes in up here, and then you have this and you mix, obviously with the sludge, you're going to mix it with polymer first. Some of the units will have a flocculating unit or a tank before it gets in. The gravity zone here that's the workhorse zone of any polymer unit. If you see, I'm going to go back a slide. On this particular one, see how big the gravity zone is right here compared to the rest of the unit, because it's really the workhorse. You're really getting out a lot of water in the gravity zone. This configuration is 2-belt, so you got an upper belt and a lower belt. So after we go through the gravity zone, it goes into a wedge zone or a low pressure zone, they call it, where one belt is starting to squeeze the sludge across these next two. This is a perforated drum, and it allows the water to get out and the filtrate to come out. And then we start to get into what your high pressure zone. So along with each set of rollers, you start to develop more and more pressure to try to squeeze the sludge tighter and tighter. Along the way, you got a belt sludge, the belts return, you got a belt wash here, you got a belt wash, and that's where some of your aerosoling comes in and kind of makes the whole area kind of moist. But they do a great job. They're very, very robust units. You change a belt, about the only thing you ever do. I mean, you got to change some belts. I'm trying to think. I don't want to say it's wrong, but maybe every 2,000 hours of operation. There's some belt spray seals here that usually get worn and need to be replaced on the belt wash, and the wiper blade needs to be washed. Or replaced. That's scraping off the sludge onto your conveyor or whatever else you have taking the sludge away. But very robust, been around, still work very well. Here's a screw press. This is a Volute screw press. There's a couple different types of screw presses. I'll talk about a Volute. They've been around, starting to really jump around into the industry lately. There's several manufacturers. This one, I stole these pictures off the internet. I probably should have fuzzed out the name like they do on TV shows. But this particular one, you have a mixed tank for polymer up here and a flock tank. Let's just jump to the next photo. I think I got it. Yeah, so we got you blend in your polymer with your sludge for prior here, so you have kind of a mixed tank. Here's your flock tank that you're utilizing. The mixture flows through these tubes. So these level probes here kind of control the level in your flock tank, your feed speed, and it flows out of the tube and then into the screw. Down at this end is your drive motor for your screw, and then there's a compression plate down here that you set manually to kind of compress the sludge to keep it from going out the screw. So along the way here, there are sets of fixed and movable plates. So the filtrate, as these plates move up and down, it allows the water, the filtrate, to get out of the unit. And as more filtrate gets out of the unit, the thicker the sludge gets, and it moves its way out and out of the unit. Their popularity, I think, has increased. It's more of a compact unit than a belt filter press. There's less aerosoling. All the water is caught within this filtrate pan down here. The only spray water that you utilize are these sprays up here to wash off the sludge that's kind of made it to the outside of the screws. There are other types of screw presses that are a little bigger devices, normally longer, and they have what they call a pressure cone that's automatically adjusted down here, and they're usually a wedge wire or a perforated plate with a screw in the middle, but they are also self-contained devices as well. So there's a couple different types. This happens to be a Volute style. So, oops, just blotted out another name here. This is a flat wig. Oh, maybe I think flat wig. They may have actually a factory here in Ohio or right across the border. So promote a little Ohio here with a flat wig centrifuge. So here, centrifuge has a main drive and a back drive motor, and I'll get into it real quick. So you got a rotating bowl and a rotating scroll, and what you're doing is kind of flinging your centrate out to the walls, and you're scrolling your sludge down to the end of and sludge is discharging. Now, necessarily, normally things go in opposite directions, or they can go in the same direction, sludge and centrate, but normally kind of see an opposite thing. Centrifuges are, I don't want to call them, but they are a black box. So once you put the sludge and the polymer together, it goes in there and you never see it again until it comes out of the unit or your centrate comes out. Now, the unit controls the torque within the unit, so it knows the torque and it's making good cake. There's one thing I would say about a centrifuge, that if you're going to haul exclusively to a landfill, the centrifuge, not all the time, but the majority of the time, will get you 2 or 3% higher cake. And the reason for that is you have now a confined situation. So as I apply more pressure to the sludge can't get away. So I can apply more pressure to the sludge. When you think of belt filter presses, you have sludge running between two belts. And if I continue to apply too much pressure to the sludge, it'll squeeze out between those two belts, and I'll lose effectiveness and I'll lose sludge. Same way with a screw press. There's only so hard I can squeeze it with the backbone or my pressure plate, or I start to squeeze the sludge out of between those, the fixed and the moving plate. So I kind of, to me, it feels like making a mud ball. If I can keep my fingers really tight and just with really tiny little openings, some of the sludge, as I squeeze too hard, the sludge or the mud will squeeze between my fingers, and it starts to get the throughput of the device gets to be less and less because I'm squeezing the sludge through those openings. So centrifuge normally always will get you the driest cake. So I'll turn it over to poll question 3.
So this question is, which of the following is not a mechanical means to dewater? You've got either of the two belt presses, single or double, screw press, centrifuge, or none of the above. We're almost at 90% voted here. Keep it going a little longer.
And Dan, I'll just mention that we'll be coming up to about 2:15 will be the final time. So we may not have a whole lot of time for questions, but just a reminder, if anyone does have any questions, we run out of time, we can also pass those along after the webinar.
I got just a couple of more slides, so I'm pretty close.
So we're at 94%, 100% now on the poll. 90% of people put in none of the above, and 11% put in centrifuge.
So we're gonna jump in. What else do I need if I'm gonna do mechanical? You need some type of feed pump. Not all the time you need a grinder. If you have good screening ahead, you don't necessarily need a grinder. I think we see grinders more often with a centrifuge and a belt press, but you can add a grinder. It's normally some type of progressive cavity style pump because you want to have an even feed rate going into the device. One other thing I don't have on this list, that pump has to draw from some type of tank, and you want to be able to have that tank be some kind of, I don't know, homogeneous mixture, whether it's your aerobic digester or anaerobic storage or sludge storage tank. You'd like to keep that tank mixed so that when you start to feed your dewatering device, you can feed it at a steady rate with a homogeneous mixture. And what I mean by homogeneous is that it's not jumping percent solids. Like if you would suck off the bottom of the tank, it might start at 3 or 4% going to the device, and then it falls back to 2 or 1.5% or something like that. That's going to kind of wreak havoc with your polymer feed and your dewatering device. So you want to try to keep it as homogeneous as possible. Some kind of polymer preparation unit. You know, there's all kinds of them out there. And then how do you want to store your polymer? Usually drums and totes are used. It all depends how much polymer you're going to need. A conveyor to get it away from your dewatering device, either a belt or a screw. You can also go right, if you set the device up high, you can dump it right into a dumpster. Again, a building to house a unit, you know, space for a backup or a future if you're ever going to need it, if you think. And then polymer, water for your polymer, and device washing, as well as some kind of power feed. So those are kind of what else you need. You know, I fall back to how often am I going to run, and how much volume do I have ahead of the unit that I need to, you know, get out of my my digestion process. Yeah, you can get a really large device and run for a very short period of time, 1 day a month, and get rid of all your sludge at once and drop the volumes down in your digesters. But that device is going to cost you a lot more money. So if you kind of weigh exactly, that's the thing, that's the price you're going to pay. Is the bigger device, the more money you're going to pay for that device. But maybe you don't have to run it very often. Maybe that meets your needs. So storage after dewatering, you know, what we see most often is, you know, we'll end up building a building on a sand, where the old sand drying bed was, and we'll dump it right back where one of the old sand drying beds was as well. So that's usually the kind of application we see quite a bit. And I think, how do I make this final decision? Visit other plants. You don't know what dewatering device you have, network. Go to local shows and meetings and talk to your neighbors, what they like, what works for them. It usually works around the industry. Contact a salesman and have them run a pilot, and have them run it for free, by the way. Usually the issue is getting on the schedule. So if you've gone to a neighboring plant and you see a device you like, hey, do you run call a salesman or two, and do you run a pilot, and get on the schedule? You may not get that pilot, you know, the next month, and maybe you won't get it till the following year, but you get an opportunity to see how that unit's going to work for you. You know, think about what are your disposal. Am I going to go both landfill and land application? Again, if you're going to go landfill only, weight costs money. Determine all your needs and capabilities, and then, you know, in the end, if you want to, call an engineer to discuss over what they've done for other communities. But I really like number one up there, network with your other local guys and see what they like, see what they've done at their plants. So with that, I'll open it up to poll question 4 or your guys' questions.
All right, so last question here. If we use mechanical dewatering devices, what other equipment do you need? You got a truck to haul sludge, a feed pump, polymer feed equipment, or wastewater, or sorry, wash water. roughly at 90% here. We'll just wait a little longer. And that was it, 100% voted. So the final results are 44% said truck to haul sludge, 11% said feed pump, 17 polymer feed equipment, and 28 said wash water.
Any questions out there?
We can give them a little time to type in here. I'm not seeing any questions at the moment.
Yeah, and we're kind of coming right up to the end of our time here. So if there are any questions that come in, that's all right, since we, you had really fantastic presentation, Dan. I know it was really, I was very happy where last week we had a TMACOG wastewater committee where we were actually able to go out to the wastewater plant in Oregon, and they have some really cool centrifuge system that they have set up. So it was really interesting to be able to see some of those things last week and then come in and be able to see your presentation this week.
Yes, Oregon has a nice setup, and they go into a couple screw conveyors and can discharge right directly into a truck or into a drum or into a dumpster, whatever fits them the best.
All right, well, I'm not seeing any questions come in. So I think we'll go ahead and I'll do the wrap up for this webinar, but just wanted to thank you again, Dan, for giving this presentation. It was really informative.
No problem. Thank you.
All right, thank you everyone for attending this second session of our virtual wastewater operator training. Just remember that for a couple of things to finish up this, to make sure everyone can get credit, the contact hour request form can be found in the handouts section of the GoToWebinar dashboard. You can download that, fill it out, and then send it back to Ann Nair, myself. TMACOG will provide contact hours to all attendees who met the minimum requirements for participation that we'd outlined previously. We'll also notify attendees of contact hours via email, and we'll be providing a certificate of attendance. We'll also upload any of the qualified attendees to the OEPA eBiz website within 30 days. And I'd like to also remind everyone that the survey at the end of the webinar is mandatory, but it will come up when the webinar is closed. Up on the screen, I have the contact information for the TMACOG staff. I also have the presenter information. You'll also receive an email in 1 hour that will also have the information for the presenters. Shortly here at 2:20, I'll be going and ending the webinar. So we don't have any issues with the participation score being messed up with people leaving at different times. But again, I'd also like to mention, so before you do that, to download that contact hour handout form so that you can get it in and everyone can get their hours. Thank you again to all of our presenters and to everyone coming by. We'll be looking to host both an in-person session and potentially another virtual session later on in the year, around in the fall. So keep your eyes and ears open for some more opportunities that'll be coming along from TMACOG, and we really appreciate everyone coming by and enjoy being able to provide these training opportunities for everyone. So once again, thank you, and I look forward to seeing everyone another time.